Hello. Now we're ready for the second portion here of the beginning of the amino acid oxidation. And so, remember we've taken, I'm going to say for example, either an endogenous protein, which is, proteins that we've already have in the cells in our body, like in our muscles or things like that, and perhaps, we're going on a starvation diet or we have uncontrolled diabetes or something, and so our body's going to try to scavenge the energy by breaking down the proteins that we already have. Or the other way is that we can just get the proteins directly from our diet. We've just taken a big meal, you've eaten that Big Mac, forget all of the horrible side effects of all the grease and the empty calories, the bad carbs. We can talk about that at some other time. But we'll just focus on the proteins for those of you who are carnivorous. And we need to break down those proteins as well. We're going to get the energy as we can. And as we mentioned in the last class, carnivores can get up to 90% of their energy right after eating a protein-laden meal from the breakdown of the amino acids. Okay? And so there are two classes of enzymes here that are very important in scavenging the energy from a protein. Okay? The first class are the enzymes that degrade proteins, whether we're talking about endogenous or exogenous proteins. And then the second class are the class of amino acids that lose their amino groups. Okay, so how do we just remove the amino groups from the individual amino acids? The first class breaks down the proteins to the amino acids. The second class is going to remove the amino group from the amino acid and turn it into an alpha keto acid. Okay? So, that first class is going to be called first class is going to break it down, because unfortunately this computer program will not let me write directly on the PowerPoint. I have to switch boards. Okay, so the first class we're talking about the proteins. Remember the tertiary, quaternary structure proteins? We're wanting to break it down into the individual amino acids. Break apart all the way through the primary structure. So what class of enzymes can do this? Remember? Those are proteases. Our good friend the proteases. Things like trypsin, chymotrypsin, pepsin, things like that. They're going to be breaking down. These are proteases. It's just going to be breaking apart the proteins into the individual amino acids. Then we have the second class, which is going to take the amino acid and transform it into an alpha keto acid. That's a C. Okay, and so what class of enzymes are doing this? This is essentially, once again, we're just going to take this. Didn't leave myself much room. And we're converting it into the alpha keto acid. Do you remember what class of enzymes it is that's going to do that? That class of enzymes is our good friend Amino Transferases. Okay? So the first class are the proteases, the second class are the amino transferases. I'm not going to go into any more detail concerning the proteases. We talked about those last semester when we talked about the hierarchy of protein structure. So let's focus on the amino transferases. First off, and perhaps the most importantly, it's reversible. Okay, we're going to really find out the reason why that's so important with respect to alpha ketoglutarate in the second bullet point. So all that they do is they're going to transfer the amino group from one amino acid to convert it into an alpha keto acid, and they're going to add it to a different alpha keto acid to turn it into an amino acid. So the the amino group gets transferred to alpha ketoglutarate. Well, every single amino transferase utilizes a ping pong mechanism where one substrate is alpha ketoglutarate. Alpha ketoglutarate is going to form what amino acid? Okay, because this amino acid is the intercessor between the biosynthetic pathway and the excretion pathway. Okay, we have to have a go-between. Alright? And so, word-wise, it's kind of difficult maybe for you to picture in your mind. I know it was for me. So I'm going to write that out, what that second bullet point means. What I'm saying here is the aminotransferase, which the old word for it was transaminase. It's going to take, let's say, I'm just going to use a very specific example. There's one called Alanine Aminotransferase. Let's say we have an alanine and we're wanting to get rid of it because, we no longer need it. We need the energy from it. Which, that's an NH3. That's alanine. It's not drawn very well. But there is an enzyme. Like I said, it is reversible. I'm going to show it this way. And it's going to convert alanine into what? Alpha keto acid. That's pyruvate. Okay, so pyruvate then can go directly into gluconeogenesis to make the energy. Alright? However, in order for it to do that, it has to transfer this amino group to another amino acid. To another alpha keto acid, I should say, not amino acid. I'm sorry about that. So what it does is every single amino transferase actually utilizes, I'm going to draw it kind of funky, because maybe then it'll be easier to understand. This alpha keto acid. What alpha keto acid is that? That is alpha ketoglutarate. And so the amino transferase utilizes alpha ketoglutarate, and it's going to convert the alpha ketoglutarate into an amino acid because what it does is it actually takes off the nitrogen and adds it in place of the ketone. So we're going to have this R group, CH2, CH2, CO2 minus. But now we have it on an amino acid. And what amino acid is this? It's glutamate. So glutamate and the alpha ketoglutarate, that's the intercessor for getting rid of that nitrogen. Okay, so it's very, very important that you recognize that. So every aminotransferase will have alpha ketoglutarate or glutamate, depending upon which direction we're going. Okay, so they are always named after the other amino acid. So this one would be called Alanine Aminotransferase. If it was serine aminotransferase, then instead of having the CH3 group, we would have a CH2OH group. But we'd still be utilizing alpha ketoglutarate and making a glutamate. Hopefully that will help sink in. Another thing is that that the aminotransferase have an equilibrium constant of approximately 1. So what does that make delta G? Okay, if the equilibrium constant is 1, the ln of 1, the natural log of 1, because remember delta G is equal to minus RT times the natural log of KEQ. Well, the natural log of 1 is 0, so anything times 0 is 0, so delta G prime is essentially 0, so essentially you're at equilibrium. They all have the exact same reaction mechanism, and they all use the same prosthetic group. It's called pyridoxal phosphate. It's figure 18.6. You need to know the mechanisms of pyridoxal phosphate. There are 3 of them. These are the 2 forms for pyridoxal phosphate. We're talking about PLP, where in the keto form, and then pyridoxamine, whenever it's got the amino group on it. Okay, and so pyridoxal phosphate, PLP, is really useful in transferring amino groups. And we'll see how in the future. Okay, so PLP is covalently attached to the enzyme. So what happens is the amino transferase, or the enzyme that's going to have the PLP on it, actually has a lysine that acts as a nucleophile, smacks into the carbon, and ultimately will replace that oxygen to form an imine. This is a Schiff base. So for those of you who've already had Organic Chem 2, you've seen imines and imine formation in the past. We're going to see this a lot. These Schiff bases are important. They are reversible, so all it has to do to take this off is a water and oxygen has to come in and smack this. It's going to kick off the enzyme. You're going to have the free enzyme, and then you'll have the oxygen replaced. Okay. There are 3 different types of amino acid transformations that utilize PLP. The one that we've been discussing so far, and that we discussed the vast majority of, are the transaminations, or the amino transferases. But PLP is also used in racemization, if you need to switch between the D and the L stereoisomers of the amino acid. And it's also important because it can be used to decarboxylate an amino acid. Okay? And all 3 types form a Schiff base in what's called the quininoid intermediate. And we're going to actually go into great detail for all 3 of these reaction mechanisms because they are very important and I want you to know them. But I'm going to cover them in greater detail on their own. Okay, but I just want to let you know that there are 3 types of reactions: the amino transferases, the racemases, which means it switches from D to L or L to D, and the decarboxylases, those that can decarboxylate. That's it for part 2 of amino acid oxidation.